Anand Kumar to introduce the speaker.
Fourth session of this auspicious conference, which is being conducted by University of Health. Lung cancer is one disease which every one of us come in contact, and everyone knows probably that the most common factor for lung cancer is smoking. Still people smoke, and as our previous speaker have very well said that one has to take one note to make the society free from any one bad habit. Or in continuum with this, I continue with this topic of discussion for fight against lung cancer. Our speaker for today is Dr. Rayos Sharma, who is working in Department of Pathology, Institute of Cardiovascular Research, VUmc, University Medical Center, Amsterdam, the Netherlands. And he's Dutch biomedical scientist and professor, affiliated to Bridge University Medical Center, Amsterdam. He obtained diploma in 1974 and further studied biological science in EMU Aligarh, obtained BSc in Delhi University and MPhil in degree of biochemistry in 2005. After a short working experience as a clinical biochemist at the Armed Forces Transfusion Center, Delhi, Dr. Sharma moved to Germany in early 1985 to the prestigious Max Planck Institute for biophysical chemistry, got in as postdoctoral fellowship in molecular biology, director Professor Tom Jobin. In 1988, he accepted a junior group leader position as MPI for experimental cardiology, director Professor Wolfgang Schäfer, Bad Neuen, Germany. In early 1993, he moved to Holland to the Rayos University Medical Center, Rotterdam, as an assistant professor and established a research group at the Cardiopulmonary Molecular Biology Lab, and simultaneously participated in medical teaching. In 2010, he joined VUmc, Amsterdam as a senior faculty. Dr. Sharma holds visiting professorship at the Leuven University, Belgium, and CSM Medical University, Lucknow, India. His area of research include role of growth factors, cytokines, and vasoactive agents in the pathogenesis of heart and lung disease. Dr. Sharma has obtained a number of research grants, published 328 peer-reviewed paper books and chapters, and has a number of cloned several genetic, including programmed VEGF and FGF1. He has more than 26 years of teaching experience of molecular and cell biology from college and laboratory sciences. He has supervised 14 MD, MS and PhD degrees, and he has been bestowed with numerous awards and medals. He is the founder president of Indian Academy of Biomedical Sciences, executive board member of Netherlands India Association Chair for Science and Technology, Council of GA, 0PIO, and member of several units. Dr. Sharma believes that scientists and the medical practitioners are not rivals but students and teachers of each other, as sharing of multidisciplinary knowledge makes breakthrough in innovation and development. I asked our first
that it will be a short talk, but I need to enhance the talk, so I'm covering the entire big area on angiogenesis.
I welcome Pathology, Institute of Cardiovascular Research, University Medical Center, Amsterdam. Sir has very prestigious awards, as I just now found from his CV, and I welcome and invite Professor Sharma for his keynote address.
Dais and my friends and colleagues of the Dais, particularly Dr. Ashok Kumar, who is known to me for almost 2 decades now, has done wonderful work on oxidative stress, antioxidants. Dr. Ashok is known for his research. When I got the invitation, I was not sure whether I'll be able to make it during this time, because this is a very tough teaching period in our university. But I managed to take out a few days and combined my travel to Kanpur. So I'm thankful to all of you, those who are organizing this wonderful conference, giving me the opportunity to share some of our work here. My talk will be covering several models based on the lungs, the tissue in the material which we obtain from patients. So this is a pure, I would not review anyone else's work at the moment. I'll present the data which we have produced over almost a decade in our lab. Just to give you a background, as far as India is concerned, there are chronic lung diseases which are associated with the lung cancer also, particularly COPD patients, they are associated with the lung cancer as well. But if you look at it, it starts as a asthma or a COPD, eventually can link to the lung cancer. But I would focus based on the data because we get the resected material from the patients, those who are undergoing lobectomy or pneumonectomy. WHO believes that the lung diseases, particularly in developing countries, are on rise, and they will be really a kind of epidemic situation in the next decade because we need to do something. We need to find better therapeutic targets. We need to develop better drugs and preventive methods. And luckily, as you might have heard, as far as India is concerned, the Deputy Director General for WHO has been appointed from India, the daughter of the renowned MS Swaminathanji. So Dr. Soumya Swaminathan has been appointed as the Deputy Director General for WHO in Geneva. And I think this is extremely high moment for India, who would definitely be focusing more on the tropical diseases, and I think the lung disease would be another a very important aspect for the country, for our country here in India. So, just to refresh your memory, what we are talking about here, as you see, in the normal structure of the lung trachea, divided into the bronchial parts, so which are, the upper part is called the conducting zone and the respiratory zone, because these bronchi are further dividing to become into the bronchioles and the alveolar regions where the gaseous section happens. So anything which is related, this is a direct touch to the environment. So anything which is in the environment would affect these bronchial tree. As you can see, if you cut in a transversely the bronchial region, the bronchus, you see that there is an internal lining, the epithelial lining, basement membrane, and also these muscle cells are embedded between these white cartilage. So it's a kind of structure which is needed to have contraction and relaxation. In patients with chronic obstructive lung disease, this structure is changing dramatically, and then in long term, this is narrowed down, and I'll show you a lot of data from our work, from our lab, what we have studied in these patients. So this becomes irreversible for contraction and relaxation of the bronchial component. You have glands, and you have lots of blood vessels here, both under the smooth muscle as well as upper part of the smooth muscle. And you have cartilage here at the bottom. In case of patients, those who are chronically sick, they develop enormous changes in the airways, which is linked to all these muscle cells are changed. These muscle bundles are grown in size, as well as they have divided. There are much more number of cells, so both hypertrophy and hyperplasia. Glands are swollen here, and also this basement membrane. So there's a lot of deposition of extra proteins which are unwanted, and also this epithelial lining is damaged. So due to this chronic situation, multiple changes would happen. The patient would become worse and worse, and eventually may not survive. So we have to find out the method, we have to find out the mechanism, we have to find out the targets, how we can really develop the new targets. So the data which I'm sharing from our lab, from our hospital, they have chronically in the gold 4 stages of patients, those who underwent surgery for lung cancer. All these 14 patients had lung function deteriorated to less than 75% predicted, means that the forced expiratory volume in 1 second is less than 75% as compared to the normal individuals. Reversibility is comparable in both the cases. However, the carbon monoxide diffusion coefficient is definitely much reduced in case of COPD patients. So there is a lung function is highly deteriorated as compared to the normals. We do have all the data related to their smoking history, pathological records, and medication. I must mention that all these patients were brought into the cardiothoracic unit where their lungs were operated to remove a tumor. So either it was entire lobe was taken away or a part of lobe was taken away. So lobectomy or pneumonectomy was done, and we were able to obtain the tissue from them to understand what was happening. So the chronic disease linked with the cancer. I would like to share several of our work, several papers I would like to summarize from our group where we have looked at the injury process. So in this piece of work we have confirmed that there is a tremendous growth happening in the epithelial layer. So this damaged epithelium is resulting into proliferation of epithelial cells in patients with chronic lung diseases. And this we have confirmed using the Ki67. It's a cell dividing or cell cycle gene. And as you can see here, it's a significant difference. So what we understood from this data is that higher is the proliferation of epithelial cell, deteriorated is the lung function, indicating that patient is really getting worse and worse. So the inverse correlation between the Ki index to the lung function data. And this is depicted here from all the patients when we compare the normal and the patient with the chronic lung disease. What we did to understand whether this repair process, because the damaged epithelium is the key, whether this repair process can be enhanced by employing certain factors. And what we did here, we took the epithelial cells isolated from the human lungs, prepared them in the monolayer, and this monolayer we created a wound. We did an artificial wound in the monolayer of epithelial cells just to understand whether we can close this wound. And what we did here, we took the fibroblast growth factor as well as fibroblast growth factor 2. Both these molecules have been shown as a mitogens for a variety of cells, but we have no clue whether it works in epithelial cells. And we did find that fibroblast growth factor 2, which is also known as basic fibroblast growth factor, is responsible for the wound closure, as you can see here in this case. And FGF1 was much less responsible for this growth. And you can plot it nicely with the time-dependent manner, and you see that FGF2 is a very clear-cut wound closer. So this is after testing variety of other factors. I'm showing mainly the positive data here. We looked at number of growth factors in the patients with chronic lung disease associated with the lung cancer. There we have a variety of things happening, which is called the vascular remodeling. So the angiogenic response in these patients is very apparent. What we found, we showed for very first time, long ago, almost now 2 decades, when I published this work was that smooth muscle cells are loaded with VEGF. And just before that, we cloned vascular endothelial growth factor from a normal tissue. As we know that vascular endothelial growth factor is apparent, and we heard a lot of other talks today as well as yesterday, that it's a key molecule for blood vessels to grow. Means that it's an angiogenic promoter. And we wanted to learn which particular cell is producing this growth factor and where we can tackle it in case of lungs, because we learned that we don't need VEGF, but in other cases we do need VEGF. So we cloned this gene, we published the sequence, we published its expression pattern in variety of organs. The data is much old now, but the relevance to our work right now is that these smooth muscle cells are the key cells, those who are producing VEGF in the lungs. So we wanted to see that the cells which are responsible for contraction and relaxation are changing their phenotype from contracted phenotype or contractile phenotype to synthetic phenotype. Means they are dividing and also producing other growth factors, and VEGF is one of them. We have also correlated the expression of VEGF, again establishing the fact that the lung function deterioration is linked with the higher expression of VEGF, so it is inverse correlation in various compartments, so the microvessels, airway smooth muscle cells, which is so apparent, and vascular smooth muscle as well as epithelial cells. So we know now in the entire lung, in different regions, the expression of VEGF is a problem, is really a problem, because if you have more VEGF, you will have more blood vessels, you will have more secretion, you will have more edema, because VEGF has also a property of releasing the fluid. So it's called the vascular permeability factor. The blood vessels becomes leaky if you have more VEGF around. We also confirmed, I've taken just one slide from one of our beautiful paper which came in the American Journal of Respiratory Cell and Molecular Biology a few years ago, that the remodeling process of the vessels is linked at different levels. So 50 to 100 micron, even up to more than 400 microns. So the smaller vessels, capillaries, to the 400 micron blood vessels, they are all getting remodeled, but the larger vessels are in problem. We established that the larger vessels, which are more than 400 micron, are becoming really thickened. They have tremendous changes. And that change, we wanted to know what kind of change it is. It is something related to the media, or it has something related to the adventitia, means the blood vessel can grow outside or inside. We established by this data in the figure B, that this is not linked with the medial thickening, because smooth muscle staining was remaining unchanged. We did number of staining, number of expression markers for these blood vessels, and you could see that in the lungs of patients with chronic lung disease, you have these blood vessels really growing within. You can see here and how they are growing, because we have done laminin, fibronectin, and variety of other factors, collagens, variety of vitronectin, number of things which we have used the markers. But they came up with the same confirmation that the medial thickening is not there, but the intimal fibrosis. So the cell type, smooth muscle cells are changing their phenotype to fibroblast, and they are growing inside the cell, inside the lumen of the blood vessels, and these blood vessels are really getting blocked, as you can see here, and they do not eventually carry the blood. And the patient is suffering from pulmonary hypertension, right heart failure, and so on, all linked with the developing cancer. So to understand the mechanism, we would like to go more deeper. We would like to find out which particular cell and the crosstalk between the cells. We know now, our work and many other labs in the world, they have established the fact that hypoxia-inducible factor is linked, it's a very early gene. As soon as you have hypoxia, that results into the production of number of other proteins, and VEGF, VEGF-A in this case, is a very prominent, linked with angiopoietin 1 and a number of cytokines and STF. And this is a key for angiogenesis, means formation of blood vessels. In a representative manner, if you can look at the hematopoietic stem cells, they are the key. They are the stem cells which will become a blood vessel. So the they aggregate and start preparing a tubular structure. They join each other. They are continuously dividing, join each other to create a tubular structure. A number of other factors derived from monocytes, platelets, and CD positive T cells. They release number of growth factors and cytokines in the circulation, and they are responsible for further division and sprouting of the blood vessels. And as you can see, and the reactive oxygen species, which is another key step in formation of a blood vessel, where there's a lot of work from Dr. Ashok's lab, also Dr. Kumar's lab, that the reactive oxygen species can result into the further perpetuation of the blood vessels, and then it will lead to the, you see this stalk cells and the tip cell. Tip cell is the one which is experiencing the hypoxic stimuli. And then again, number of growth factors are produced. So the hypoxia, and so it's a kind of chain. It's a wheel which is continuously, and then you have this blood vessel form. And that's what's happening in the lungs of patients with a chronic lung disease. So we have to do something to reduce this formation of blood vessels. We do not want angiogenesis happening in the lungs, because if you have more angiogenesis, you will have more edema and the patient would be in trouble. I would like to go into more and more deeper. So the whole work which I am presenting is coming from patients, means the bedside. So the patient has a trouble. So from bedside to the bench side, we brought it to the pathology lab. Now we are bringing from pathology lab further down to the molecular level. We would like to identify the cell crosstalk, the genetic manipulation, understanding the mechanism. So we are going from isolating the cells, particularly cells. As you heard that in my previous few slides, that the VEGF, which is the key molecule, is produced by smooth muscle cells. So we decided to culture them, and these muscle cells are embedded in the cartilage. So it's a very difficult and very delicate situation to isolate and put them in culture. We established over many years, and you need to, we were getting a very small piece of biopsy. From that we have to isolate the cell. We put them in culture and grow them for several weeks. And then comes the experiment. And how we did it here, you can see this little piece of muscle. We scrape the epithelial layer, we cut out and dissect out the smooth muscle. We put that smooth muscle, tiny pieces, into the condition, into the medium, and put them in culture, and these cells, they grow as a sprout, as you can see here. And then these cells are really characterized. You could do alpha smooth muscle actin staining, and then you would see that they are very pure. Now we can do a lot of experiments. We can play around by treating these cells with number of growth factors. We can also understand the artificial model by which we can understand the mechanism of breathing. So this is one of our first experiments where we did the cells, isolated, cultured them for several weeks. Then we serum deprive them. So you need at least 3 million cells per time point. So we need to have enough cells to do this experiment for messenger RNA and also for protein. We serum deprive them for different time points and then treat them with number of growth factors, cytokines. We treat them also azithromycin, another macrolide which is available routinely and you use for the patients routinely in the clinic. And then we wanted to have the end point after treatment, what kind of molecules are being produced by these cells. So TGF-beta is one of the key molecules which is responsible only for the matrix formation in the lungs. So we wanted to see if we have more TGF-beta. We showed about in one of our early papers that TGF-beta is a lot present, and this TGF-beta is responsible for the formation of the matrix. So we wanted to see whether this matrix formation is linked with the VEGF production. So the vascular endothelial growth factor is several-fold increased here, and as you can see with the control. And if you measure the protein from these cells, from the conditioned medium, the medium which you collect from your cells, you see that this is a time-dependent but accumulation of protein, which is 24 hours, is already very significant, and obviously at 48 hours, the very high protein is in the conditioned medium. We wanted to see that the TGF-beta induced VEGF produced in the airway smooth muscle cells is biologically active. For that, what we did, we treated the cells with the inhibitor. It's an inhibitor which blocks the VEGF receptors on HUBEC, human umbilical endothelial cells, and these human umbilical endothelial cells will not proliferate if that was our hypothesis. And indeed, that's what it happened. So we take the conditioned medium, we see that these cells are dividing here, and when we have the receptor inhibitor treated cells, this division is stopped. So we could establish with this experiment that TGF-beta induced VEGF is biologically active, and it's all happening in vitro. We did many other molecular events to understand the signaling mechanism, and here we established that ERK1 and 2 MAP kinases and also P38 MAP kinases, they are also linked because they get phosphorylated in response to fibroblast growth factor, and if we treat them with azithromycin, you are able to block the not P32 but ERK, and as you can see here very clearly, and here also. So we know that these growth factors are responsible for inducing the signaling mechanism via ERK pathway. And this ERK pathway, whether it is linked with VEGF, is our next experiment. We would like to know that these both, dexamethasone has been used. These are the all steroids we use in the patients. Also this macrolide azithromycin is used. And interestingly, these existing drugs are responsible by reducing the VEGF production, which is FGF1 or FGF2 induced. So both these growth factors induces VEGF, and we can block it. We can reduce this production by azithromycin. So existing drug, finding out a new property. We also looked at the role of nitric oxide synthase, whether the nitric oxide is responsible for VEGF production. We are trying to find out the targets, and here we have all the cells, the airway smooth muscle cells, well characterized, and also pulmonary artery endothelial cells, and also they are characterized with CD31. What we did here, we took the airway smooth muscle cells, treated at different time points with IL-1 beta. IL-1 beta is a cytokine, induces VEGF. We could block the production of VEGF by L-name. L-name is inhibiting the nitric oxide synthase, means NO production. But when we put L-arginine, we see that this is reduction is restored, means the L-arginine is a nitric oxide donor, and at different time points, 4 hours and 24 hours. And when we measure the protein, it is well described here that we are able to block the protein when we are able to block the using the L name, means nitric oxide synthase, means the enzyme which is responsible for production of nitric oxide. So there is a synergy between nitric oxide and VEGF. So another target we found in the lungs, and we could establish this also by using the protein, VEGF protein at 24 hours, very well. Here it is reduced. IL-1 beta induces, but when we use L-name, it is completely blocked. But when we use L-name as well as with L-arginine, arginine would have a nitric oxide donor properties, means that VEGF is restored. But we also have looked at another molecule which is the family of VEGF called placenta-derived growth factor that was also in a similar fashion reacting. So what we understand from nitric oxide hypothesis, which we can really put this two cell talks together. In smooth muscle cells, IL-1 beta induces nitric oxide synthase, it produces nitric oxide, which is a vasodilator. That in turn induces VEGF, and this VEGF, which is produced from the smooth muscle cells, bind to endothelial cells and is responsible for angiogenesis. So if we have a possibility to block this VEGF production from smooth muscle cells, we can also block the angiogenesis. So nitric oxide pathway is another possibility. But again and again, all these targets which I'm mentioning, we have to be very careful because we are talking about heart and lung. They are both work in a different manner. In the heart, we want to have blood vessels healthy, we want to have blood vessels growing, we want to have blood vessels existing or neat and clean blood vessels. So if we are blocking the blood vessels in the lungs, we have to have a targeted therapy. We have to have only to the lungs, not to the heart. Lastly, I would like to elude some more work from our group where we have looked at the cyclical stretch. So we thought we can create an in vitro model in the lab, and this in vitro model we can use to understand the mechanism, whether the mechanical stress is responsible for also at the genetic manipulation which are happening in the cells. So for this, what we did, we took the cells and cultured on the 6-well plate cystic membrane and applied vacuum in a cyclical manner. So the cells are grown like this, and then you have a vacuum applied here. So the cell down and released. So it's exactly like in the lungs when we are breathing like this. Here we have an exact situation where we are trying to create a stress. So cyclical strain to these cells in vitro. So we wanted to create a model of chronic lung disease as we see in the patients. So we apply the cyclical stretch to the patient, to these cells grown in culture, and we would like to use that in a fashion by which it's comparable. So about 15 cycles, we use 15 cycles for these cells. And we applied the uniform biaxial stretch to these patients. And this was the experimental setup. We have the human airway smooth muscle cells, cultured them, put them in culture. We serum deprive them to bring all the cells at the same level. Then we apply the uniform biaxial stretch. So it is not exactly 3-dimensional, but 2-dimensional situation. But I think we can understand a lot whether this mechanical strain in a cyclical manner is responsible for the disease process which we have. And we did a lot of things here. We decided to do the antibody array, like we have in genomics, we have DNA arrays, like we have also now the antibody array. So we use the antibody array. Here you can have selected antibody array. So we took mainly the cytokine array with variety of growth factors and cytokines. So 88 spots on one filter. As you can see here, in control without a stretch versus the cells, those who underwent mechanical stretch, you see there number of spots have come up, and they are much more prominent. And these new spots which are coming up, they are all due to the mechanical stretch. So we can quantify these proteins and then plot them here from different arrays, and then you can see the angiogenin, VEGF, and GROW. Number of really responsible molecules are coming through the stretch. So this model turned out to be a very beautiful model to understand whether the mechanical stretch is responsible for production of cytokines and growth factors, and this is very apparent, IL-6 and IL-8 and so on. We continue this work to link whether the VEGF which is produced here is due to the stretch is associated with a pathway, is a signaling pathway. And here we did again P38 and ERK. So the phosphorylation due to stretch, and we can plot it nicely. So it is a transient phosphorylation of ERK as well as P38. We can measure by ELISA the protein, and when we look at three different inhibitors, Wortmenin, U0261 and Y27632, this is P38 linked with RhoA, and then here is the ERK pathway and the Wortmenin. Only one pathway is responsible for reducing the VEGF. So out of these many experiments, we could establish that VEGF production is reduced when we block the ERK pathway. So out of these several experiments, we could exploit in future, and the work which is underway, we can exploit it in terms of ERK pathway, the MAP kinase pathway. We can also use the nitric oxide pathway. And we know now that the mechanical strain is responsible for such kind of events in the lungs. It's eventually all linked via the growth factors. Now to summarize everything, what we learned so far, it's a very complex situation, but we know a lot. What we know here is that this dysregulated situation of epithelium, which is getting damaged due to the noxious particles and a variety of things which are being exposed to the epithelium, is the trigger. It's an initial stage, very early stage, responsible for this dysregulated repair mechanism. And under that we have number of cells producing growth factors are coming. They are being brought in there, and the production of the VEGF and other molecules by these airway smooth muscle cells is the key. So the exposure between the environment underneath, much below smooth muscle is the key. So we have to avoid somehow these smooth muscles to get exposed to any growth factor or any inflammatory cell, because otherwise they change their phenotype. They change from contractile to synthetic, and they start producing all sort of factors. And then you have the remodeled vessel, you have the airway is also terribly getting narrowed. This muscle mass is created which is more hypertrophied. There's a deposition of a lot of matrix proteins, and the situation becomes complex and complex. So in summarizing this, in patients we need to have several targets which our work has indicated, and obviously many companies have taken over from that the indications which we have produced through this work. And I would quickly like to thank all my students, those who contributed, the funding agencies, the Heart Foundation, also the Asthma Research Foundation of the Government of Netherlands, and also two of my excellent surgeon colleagues, those who are collaborating with us to provide the human material where we can directly work with the human tissue and really play around to understand mechanisms and to finding out the further details which are happening. And obviously, so Ed Bogers and Peter van der Spek as excellent collaborators. I would like to thank all of you for listening me, and do visit sometime if you get chance to come to Netherlands. It's a beautiful country for flowers. We have in April, about 6 weeks, these amazing fields. The entire country, middle of the country looks like this. Wherever you see all variety of colors, and the windmills of 17th century. The country is below sea level, about 38% is below sea level, but it's very beautifully managed through dikes, blocking the sea to come into the country land. Thank you very, very much.
Work on molecules in such a manner that probably anybody who has worked in fields of lung cancer and COPD and asthma can understand that this is the one molecule where we should target just to prevent the disease from emerging rather than treating or before it emerges. And I really appreciate the presentation by you, sir. And
I invite Mr. Next talk is Mauritian edible mushrooms as nutraceuticals in cancer chemoprevention. Speaker is Dr. Srishti Ramsar from University of Mauritius. Program Director is Dr. Sipra Mathur, Senior Dietitian, Kanpur Nagar. And chairpersons are Professor Alak Kumar Singh and Dr. P.P. Pidarsi.